Thanks so much for joining us today. My name is Emily Knight. I am with the LENFEST Ocean Program. For those of you that don't know us, the LENFEST Ocean Program is a grant-making program that funds research projects and expert working groups to address priority science needs facing ocean and coastal decision makers. To learn more about us, please do visit our website at lenfestocean.org. You can also sign up for our newsletter there and follow us at LenFestOcean on Twitter and Instagram. So for today's agenda, we are really excited to have Dr. Jeremy Sterling along with members of the research team to share information with all of you about a project we're supporting to examine the relationships between northern fur seals, pollock, and climate change. This project got underway in early 2018 and will span the next couple of years. So just some webinar logistics. We do have all the attendees muted. With so many people on the line, this is just to prevent feedback and echoes. We will have question and answer at the end of the presentation. To submit a question, use the Q&A panel to type your question, and I will read it aloud at the end for the researchers to answer. And you can submit your question anytime throughout the presentation. We'll keep track of the queue and read them out at the end. Depending on how many questions there are, we might not get to them all, but you know, we encourage folks to definitely follow up with us. For next steps, we are recording this webinar. In the next couple weeks, we will make the webinar recording, the PowerPoint, and a short webinar summary available on our website. Once up, I will distribute all of that information. We now have a running distribution list for information about this project. If you are not on that list or not sure if you're on it, please do reach out to me, eknight@pewtrust.org, and I'm happy to add you. Also, feel free to reach out if you have any broader questions about the LENFEST Ocean Program. You can see my email also on that first PowerPoint slide, along with Jeremy and Yvonne Ortiz, who's part of the research team, so we are all very, very easy to find. So at this point, I am going to turn it over to Jeremy Sterling. Jeremy is a research biologist with the Alaska Fisheries Science Center and is an expert in fur seal ecology. So Jeremy, do you want to take it away?
Yeah, thank you. Are you hearing me, Emily?
Yeah, I hear you fine.
Great, all right. So first, I'd like to start out, I'd like to thank both Emily and Victoria and the folks at LENFEST for giving us an opportunity to talk about our project. And also, I'd like to thank all the attendees that are online today for taking the time out in the middle of a Wednesday to listen to this talk. And the main reason for this talk is an outreach effort for us to inform the various stakeholders and other researchers that are working in the Bering Sea and the general public. And so this project, just like Emily stated before, it's just started. It started February 24th. This isn't a results-oriented talk. We just, like I said, started the project, and we're in phase 1 of the bioenergetic modeling. The title of our project, you can see there, is using bioenergetics and spatial data to quantify how northern fur seals interact with prey fisheries and climate. And this is a collaborative project between the Joint Institute for the Study of Atmosphere and Ocean at the University of Washington and the Resource Ecology and Fisheries Management and Marine Mammal Laboratory at the Alaska Fisheries Science Center, and it's supported by the LENFEST Ocean Program. I also want to say that this project couldn't be done without the continued support. This is additional support for the development of the Seattle feast and A-climb models, and we really appreciate that continued support. So first, next slide, Emily. I'd like to introduce our research team. Nick Bond is with JASAO and PMEL here at AFSC. His research focuses on Bering Sea climate variability, atmospheric forcing, and air-sea interactions. He's also the Washington State climatologist. Kirsten Holzman, she's with the REAM group here at AFSC. She developed the Seattle model, which is a multi-species stock assessment model. It's what we're interested in getting fur seals into. And she's also part of the A-climb team. Elizabeth McKieran, she's with GESAO now, and her research focuses on marine mammal energetics with fitness and population-level consequences, and she's going to be developing the northern fur seal bioenergetics model and be speaking after me. And Yvonne Ortiz, her research focuses on ecosystem approaches to fisheries management. She's a food web modeler and one of the developers of the FEAST bioenergetic model of the Bering Sea. Kieran Aiden is also part of this group. He's a program manager for REAM, is really involved in ecosystem-based fisheries management. He's a food web modeler and also is one of the developers of the FEAST model. And myself, I'm Jeremy Sterling. For those of you that don't know me, I've been working on Antarctic and northern fur seals ecology and demography for the past 22 years. Next slide, please. So this project's going to be setting out to address these 4 questions. The first question is, what are the energy requirements of northern fur seals in the Bering Sea? This isn't a new question. It's been addressed several times, dating back to the days of Elliott in the late 1800s, up to more recently. There's been some nice work done at UBC with Andrew Trites and Dave Rosenblad. The second question we'll be addressing is, what is the prey size, species and size allocation needed to match those estimated energy requirements? The third question is, what are the climate-specific northern fur seal-based multi-species harvest rates for eastern Bering Sea pollock, given the observed spatial and temporal relationships between fur seals, their foraging patterns and estimated predation rates, and pollock availability? And our last question is, what is the expected future availability of pollock and its potential impact on northern fur seals? Next slide, please. So I'm going to start the talk. I'm going to give a brief overview of the project motivation, some of the existing field work that's helping with some of the inputs of the bioenergetics model, and past data that we've collected on northern fur seal ecology. Liz McKieran will follow up my introduction and describe the bioenergetics model, and Yvonne Ortiz will discuss the FEAST and Seattle model implementation. Next slide. So the reason many of us are here is for folks that have been working in the Bering Sea, they're very familiar with this decline. What I'm showing in this figure is the eastern stock production. And when we say eastern stock, that's a management unit, and it's a management unit that's comprised of 3 islands where fur seals breed in the summertime in the Bering Sea. St. Paul and St. George, which are part of the Pribilof Islands, which are shown as the black dots in this figure, and Boguslav Island, which is shown as the purple dots in the figure. And if you look at this slide, starting at the left, the peak of the pup production, the pup production is an index of the overall population size. You can see that the first part of the decline that's highlighted in gray was largely due to an adult female harvest. This is a harvest in an attempt to increase the reproductive rates of females, but it didn't work, obviously. And so the first part, 70% of that decline is attributed directly to the removal of females. The part this project's really interested in is this unexplained portion of the decline, the area highlighted in blue, right around in the 2000s. And on, most of that decline that you see there with the black dots is directly attributed to losses we've seen on St. Paul, which is the largest aggregation of northern fur seals. The red dots are the eastern stock as a whole, and you can see the rate of decline is not as fast as just the Pribilof group, and that's largely due to the increases, which are shown as the purple dots that we've seen at Boguslav Island. And in addition, we've seen the numbers on St. George stabilize beginning in 2004. Okay, next slide, please. So this figure that's in the middle, what we're seeing is that the demography, or this pup production, is aligning with the summer foraging grounds and diet complexes. The colors depicted in this figure represent each of the islands, St. Paul, St. George, and Boguslav. St. Paul is shown as the green, or the dark blue dots and the turquoise dots. Those represent the east and west side of the island. St. George is shown there as the pink and purple, and Boguslav Island is shown as orange. And what we've seen, about 80% of the foraging during the summertime is done on the shelf, and the shelf is depicted as the kind of the red polygon lines you see there. The left side of that polygon is the shelf break, and to the left of that, the water gets really deep, down to about 3,000 meters. So about 80% of the feeding from St. Paul is in that polygon area during the summer, whereas on St. George, it's split between the two islands, St. Paul, sorry, not between two islands, but between spending time in the basin and spending time on the shelf. And the foraging that goes, the animals foraging from Boguslav, all these would be adult females, is in and around in the deep basin area. So there's strong, what we see is strong segregation during the summertime. And when these animals leave for their winter migration, which is not shown in this figure, they basically mix throughout the North Pacific and Bering Sea. And so our running hypothesis is that this summer foraging and pup provisioning is important to the overwinter survival for both pups and adults. And I'll also say that about 80,000 pups were born on St. Paul in 2016, 20,000 on St. George, and 27,000 on Boguslav. So there's more pups born on Boguslav now than St. George. Next slide, please. So, and if you remember in the previous two slides, there were differences in terms of the production. This was investigated in 2005 and 2006. An NPRB-funded study funded the COF study, which set out to examine the differences in feeding behavior and pup growth between the two islands. And what you're seeing here is a figure that shows the trip durations. And these are trip durations of moms that have given birth to pups, and they're going out to sea to replenish their dietary needs and also come back and nurse their pup. And while they're away at sea, the pup is fasting on land, so they're not getting any other resources. And the main difference here is that the trips on St. Paul Island are, in some cases, 3 times as long as what we see on Boguslav. Next slide. Looking at this another way, in the same study, a COF study comparing the two islands, where we see these differences in demography and also trip durations, we see differences in how they're able to continue to store their fat on board. So the left figure and the right figure both depict measurements of body fat on adults, which is on the left, and pups on the right. And the colors on the left are the different islands. Orange is Boguslav and black is St. Paul. So in focusing on the left figure, these are adult moms that have shown up on Boguslav that were measured for the amount of fat they had on board shortly after they gave birth to their pup, compared and then recaptured later in the season in October, and their fat was measured again. And what we see with Boguslav in the 2 years, this is in the mid-2000s, is that they pretty much maintain their fat stores throughout the season. This is in contrast to what we see on St. Paul Island, where those females that were measured in these 2 years were losing anywhere between 25 and 35 grams a day. Now, if we switch over to the right figure, the colors now depict the sex of the pup, and from the beginning of the season, birth, and then late season October. And what you can see in the left part of the right figure under the birth column is that the St. Paul and Boguslav pups are in similar condition, or they have similar amount of fat stores. This supports our winter migration work. Many of these females are feeding in similar areas from the 2 different islands, and they're coming back in similar condition. But by the end of the season, what we're seeing is those Boguslav pups have a lot more fat stored, and they're significantly different males and females from those in St. Paul. This is also, they also have much higher growth rates as well, which is not shown on Boguslav Island. Next slide, please. So why is trip duration important? One of the big parts of this project is aggregating other studies and finding information within those studies that may not have necessarily been published. These are observations from Mary Donahue's PhD thesis, where she looked at pup energetics growth and she monitored the mom's trip behavior. And so it's an example of good pup growth, which is on the left and depicted as the black line in each of the figures, versus poor pup growth on the right. Okay, so let's start with the left figure. We can see the pink dots represent the trip durations of the mom. Again, and her study was done on Reef Rookery on St. Paul Island in both years. With the figure on the left, the trip durations of the mom of this particular pup were short. They were anywhere between 4 and 6 days. They were consistent throughout the season, and as a result, we saw consistent growth in that individual pup. And if you switch over to the right figure, what we see is that when those trip durations are extended, in this case, the first 3 trips of the mom were anywhere between 8 and 11 days. The pup actually began to lose weight through the season, and then once the female honed into some good foraging grounds, her trips became shorter, and then you can see the subsequent pup growth. The integrated effect over the season is that the pup on the right had much lower growth per day compared to the pup on the left. And we believe this is in relation to the availability of resources for these 2 different moms. Next slide. So how variable is the trip duration from year to year? There's a lot of data that's been collected on this, and we've spent some time digitizing and putting together. This is a combination of historical observational trip duration studies and more contemporary telemetry studies that just demonstrates that the trip durations of moms provisioning their pups can be quite variable from year to year, ranging anywhere between 4 and 10 days. And we want to understand the energetic consequences of this variability, as well as identify what might be driving that trip duration variability. And next slide, please. And again, we think this variability is reflected in the food availability. This is work from Mike Goebel's PhD thesis. He's given us all his data from his thesis. He had a very intensive foraging. He did a very intensive foraging ecology study looking at adult females, measuring their trip durations, diving. There's over 1 million dives recorded, the female's diet, and subsequent their pup growth. He also collected close to 50 field metabolic rates on the adult females. What you're looking at here is the results from 65 seals where their enemas or their scats were collected. If you look at the right figure, the pie chart, it gives us kind of an overview. What we see is a consistent pattern that pollock and squid are the primary resources for the individuals that were sampled in this particular study. If you go to the top left figure, the nice thing about Mike's work is the collection of these diet samples really shows the changes in pollock size through the season. So those 3 histograms show the size distribution of the samples, the pollock samples, H0s that were collected. And by the end of the season, those individual fish or those individual pollock can have a 4-fold increase in energy compared to the earlier part of the season when the first eels first arrive. Next slide, please. So in addition to the trip duration variability, we also see year-to-year difference in the dive behavior. We have over 17 years of dive data, and we've recorded over 4 million dives. And the dive depths that occur on the Bering Sea shelf tend to align with the age-related behavior of pollock, becoming more demersal with age. Beth Sinclair and Roger Gentry showed this from their work in the 1980s. Beth showed by looking at pelagic scientific stomach samples that were collected in the early 80s that there was a change in the dominant age structure of pollock that was in the stomachs that coincided with strong year classes at the time. And Gentry also at the same time showed that dive variability, deeper dives versus shallow dives, aligned well with Beth Sinclair's work. And so because we see this in our 17-year time series of diving, Carrie Kuhn put together this nice fine-scale first seal foraging ecology study by using autonomous vehicles and accelerometry and video. And the autonomous vehicles are sail drones, and they carry echo sounders, and that's the echo sounder data is depicted in the figures on the right. The echo sounders, the fishery folks are able to identify pollock in the water column and get an idea if it's older pollock or younger pollock. And in this case, if we look at the figure on the right, there's a backscatter layer at about 20 meters. This is age 0 pollock near the thermocline. And the sampling, this echo sounder sampling, was done in and around where a fur seal mom was foraging, and the black line is her diving, and you can see that she's diving on that age 0 pollock. So the shallow dives are shown here as feeding on age 0s. If you contrast that to the bottom right figure, those kind of upside down V's are larger pollock depicted in the echo sounder, and the female that was in the area where this larger pollock was diving down to the bottom. So these results were really interesting, and we wanted to actually see what the animals were eating to further verify this variability and association with different sizes of pollock. Next slide. And hopefully you guys can see this out there. We were able to put some cameras on these individuals. In addition, at the same time, the sail drone was measuring the available prey field, and we also recorded all the different measurements of diving and feeding on the shelf. So what you're seeing here is the cameras on the back of the fur seal, and her head's coming in and out of the screen, and she's swimming through a large school of age 0 pollock near the surface. So this video depicts the diving information and echo sounder information we saw on the previous slide. Get a little vertigo watching this. And if we contrast that to the next slide, please. This is another female. She's diving deep. She's going down to the bottom. This is a really neat video. If she gets down to the bottom, you'll see some brittle stars and an unlucky pollock that happens to swim right in front of her. And I'll let that go again so everybody can see that. So the nice thing with this camera work is it's providing us a lot of information. It's validating some of our assumptions, and it's allowing us to use a long time series of diving information. It's also giving us some idea of the foraging efficiencies with these individuals, because we can see dives where they don't catch any food, and we can see dives where they do catch food. Okay, next slide. So to kind of wrap up my end of the talk, I'd like to say that, over the past decade, our group here, the Marine Mammal Lab, has been aligning historical data collections with contemporary studies. We've really been involved in standardizing our telemetry archive, both of location and diving information, as well as the diet collection, so we can compare studies across years. We revisited, revived, and reanalyzed previous studies and unpublished data that are really relevant to developing the bioenergetics model. And we've identified gaps and initiated fine-scale studies to kind of fill those gaps, knowledge gaps. All this is in an effort to get the eastern stock and northern fur seals into ecosystem-based fishery management modeling framework so that they can be included in the AFSC's multi-species stock assessment model, or Seattle model, and the FEAST bioenergetics model of the Bering Sea. Next slide. And so our project goal is to combine a spatially explicit fur seal bioenergetics model with the ecosystem and stock assessment models, so we can provide feedbacks between pollock and fur seal stock assessments and contribute to conservation goals. And with that, I will pass the mic to Elizabeth McCarran.
Emily, can you hear me?
Yes, I can.
Okay, thanks. Okay, so I'm going to start off talking about sort of the first phase or question of this project, which we are currently working on right now, and that is, what are the energy requirements of northern fur seals in the Bering Sea? Next slide, please. And to do this, we're going to be using a bioenergetics model, And bioenergetics is simply the study of energy flow in living systems at any level of biological organization. So, for example, it could be at the level of the individual, the population, or even species. And bioenergetic models really just help us understand how much energy individuals or populations are needing and how much food they consume to meet those needs. And these have been used for a variety of species, from anything as small as zooplankton up to fish, all the way up to these upper trophic level predators like northern purse seals. And they can even be used to model energy flow within food webs or entire ecosystems. Okay, next slide, please. So for those of you that may not be familiar with this bioenergetic model approach, I just wanted to provide a very brief introduction using a simplified example, using myself as an example. So for example, here on the left-hand side are some potential energy needs for myself in any given day. So you can see that these are broken up into different categories. So for example, time spent working at a computer, time spent running errands, exercising, and sleeping. And for each of these activities, we have sort of the energy that I'm expending doing each of these activities, which is based on the time that I spend doing that activity and also just the rate of energy that's expended on that activity. So if we take those across the entire day and we sum those up, we might say that I would need about 2,100 calories in any given day. So that's my energy need or requirement. And if we knew my diet, so theoretically, let's just say that my food consumption consisted entirely of spaghetti. And we knew the energy content of spaghetti, so for example, the number of calories per gram, we could calculate that on average, I would need about 3 pounds of pasta per day. And that's just to break even, so that's not to gain or lose any weight. So that's just a very simplified example of what a bioenergetic model might look like. And so I think the underlying concept of this model is very simple, but for marine mammals, putting these bioenergetic models together is a bit more complicated. And that's primarily due to challenges associated with quantifying not only the activities that animals are doing and how much time they're spending in each activity, but also the amount of calories or energy that they're expending doing those activities. And so as Jeremy noted previously, we aren't the first group to attempt to create a bioenergetic model for fur seals, but one of the unique things about this effort is that in the past, the bioenergetic models have been relatively simplistic, and that's primarily just due to lack of data. And so we really have this wealth of data available to us, both from studies on wild animals and a lot of more recent work on captive fur seals that can help us develop a more complicated and hopefully more biologically realistic bioenergetic model for fur seals. Next slide, please. So what does our bioenergetic model look like for fur seals? So we're actually going to be developing a number of bioenergetic models for the different demographic groups. So one of our primary focuses is on adult females that have pups, but we're also interested in quantifying the energy requirements of the population as a whole. So we're interested in other demographic groups as well, such as adult males and juveniles. And there's really 2 primary reasons for splitting these bioenergetic models among the different groups. And the first is that these different groups might have different energy. So for example, an adult female will not only need to provide for herself, but she also needs to provide energy to her pup, whereas juveniles don't have any reproductive energy requirements, but they may be more heavily invested in putting energy towards growth. And then the second reason for splitting these groups is because the behavior also may differ among, adult females versus juveniles. So, for example, adult females are going to be really tied to the rookery, so they're constrained in how far and how long they can stay away because they need to return to nurse their dependent pup, which is going to influence the amount of time that they spend foraging at sea, whereas males and juveniles don't have the same constraint, and so they may spend longer at sea and also may go to different locations. And so the overall approach we're using for the bioenergetic model is we're going to be using a simulation approach, which simply just means we're creating a population of fur seals, and we're going to simulate their energy requirements. And we want to do this actually for specific targeted years. And I'm going to talk a little bit in the next slide about what those years are going to be. But it's given sort of the behavior and pup growth rates that we observed in those years. And so, for example, what we're going to be doing is we're creating our population of fur seals, and then we can simulate them arriving and departing from the Bering Sea using sort of these known patterns that you see on the left-hand side, the lower left-hand side, of arrival patterns of these different demographic groups. So typically, adult males are arriving first, followed by adult females, and lastly by juveniles. And then once they're in the Bering Sea, we'll be simulating them going out on foraging trips and estimating based on the amount of time they spend at sea, how much energy they're expending, and whether or not there's other energy requirements they're incurring at the same time, for example, energy for growth or to put into a pup. And so what this approach really allows us to do is it allows us to incorporate the fact that animal behavior may differ throughout the season, and it also allows us to incorporate the fact that pups are growing throughout the season, and so the energy that a female needs to invest in her pup is obviously going to increase as the pup gets larger and larger. Next slide, please. So we have 6 initial target years, and we're really focusing on these 6 years that you see here on the top for 2 reasons. One, we have, these are years in which we have high-resolution data. So we have either high-resolution data on behavior and or pup growth rates. And the second is that there was variable environmental conditions among these years that influenced both female behavior and also pup growth rates. And so on those 6 panels on the top, what you can see is those are tracking data from adult females from a single rookery, the same rookery in each year. And the red versus yellow colors simply correspond to intensity of use. And so the primary purpose of showing this is just hopefully for everyone to see that animal behavior where animals forage differs among the years, which could potentially influence the energy requirements. And then on the bottom, the figure on the lower portion of the slide just shows the pup growth rates in a subset of these years. And we can see that male pups are shown here in black and female pups in gray. And the dashed lines simply correspond to the pup growth rates on Boguslav. So what we can see here is that there is variability in pup growth rates among these years, but that in all of these years, pups are still growing less than they would if they were on Boguslav. So overall, starting with these target years is going to allow us to really understand how energy requirements are affected by animal behavior and also variable investment in pup growth. And the overall goal with starting with these initial years is hopefully to end up with the ability to extend the model out to other years where maybe that maybe aren't quite as data rich. Next slide, please. So I talked briefly before about we have this wealth of data available to us to parameterize our bioenergetic model. So I just wanted to touch briefly on what kind of data we're actually going to be using in the model. And so what you're looking at here is just some of the input parameters for the adult female model with pups. And Jeremy mentioned a few of these studies earlier, but there's been a number of targeted studies on pup growth rates. So these studies were actually ones that tracked, individual pups throughout the season to look at how pup growth rates changed. We also have a targeted study in 1995 and 96 that actually quantified the milk energy intake of pups throughout the season. So not only do we know how much energy pups are taking in, but we also know how that changes as they grow. Jeremy also mentioned that these data are unpublished, but we have estimates from 48 females on at-sea metabolism. So these are free-ranging females where we know how much energy they were expending at sea. And then we have other behaviors such as all the at-sea behaviors, understanding, how long trip durations are, how long females are spending on shore with their pups, and then a number of pup parameters such as how much pups weigh at birth, et cetera. So we have a lot of data there. And then the second component of this is really sort of the demography component. So there's been long-term population monitoring studies on the Pribilof. So, and we really need this data in order to say, how many females or how many pups were born in our target years. So that's going to be necessary because we need to know how many females to simulate in our models. And it also gives us information about the distribution of animals around the island. And that's really important because, as Jeremy touched on earlier, animals that are in different locations on these islands are going to be foraging in different locations in the Bering Sea. Next slide, please. So that really leads us into the second question, where we're really interested in taking the output from question 1, which is really the energy requirements of both individuals and the population as a whole in these target years, and translating that into prey biomass, and in some cases even understanding how that relates to sort of specific consumption of different size and age classes of fish species. Next slide, please. So you've seen these figures before, but in order to understand how much prey a first seal is eating, we need to both understand its diet, which is the pie chart shown on the left that Jeremy showed earlier. And so what we can do is we can take our output from the bioenergetic model and combine that with estimates of both the proportion of the diet that's comprised of a particular prey species, and also what we know about the energy content of each of these prey species. So, for example, how many calories per gram is a pollock. And using those two data, we can estimate the biomass consumption of the primary prey species for each of these years. And then the second component of this is for specific species, and mainly for pollock, in the years where we have a good understanding of the sizes of pollock that were being consumed, we can actually estimate the age-specific consumption of pollock and actually the numbers of each of those age classes that were being consumed. Next slide, please. So so we're sort of in the midst of this bioenergetic modeling approach, but we just wanted to provide a brief example of what these numbers might look like. And so we actually ran our bioenergetic model for 1974, which is not one of our target years, but we chose it because we had some diet estimates that were readily available. So if we use the behavior of females in 1974 and the pup growth rates, the output of the bioenergetic model would suggest that the average lactating female consumed about slightly over 1 million kilocalories across the entire foraging season. And now the estimates of diet aren't that different from the pie chart that you saw earlier, suggesting that pollock comprised a large proportion of the diet, but that ganeta squids were also important. And then there's lesser importance of things like Bacillus, et cetera, in the diet. So, for example, if we take our 82% of the diet comprised of pollock, and if we assume that that directly relates to the energy consumption, so for example, if 82% of the diet is pollock, we assume that 82% of the energy is coming from pollock, we can translate that into an estimate of biomass. So here we would estimate that the average female consumed about 750 kilos of pollock in 1974. And we did have size distributions available for that year, and that would translate to about 13,000 age 1s, which was the primary age class that was being consumed, and about 168 age 3+ pollock. And so we can do that for each of those species. And so this will really be the output, an example of the output from the second phase of this project. Next slide, please. And then the last part of this phase is really, from the first part we understand how much energy they're consuming. The second part is, how that translates to biomass. But then what we really want to know is where is this energy being consumed in both time and space. And so we can do that by combining all of the tracking data that Jeremy mentioned previously and the diving data and combining it with the output of the bioenergetic model to really come up with a 3D energy consumption landscape, which is what you see on the right here. And so here on the one axis you have depth, and then you have longitude and latitude. And primarily what you're seeing here, the way the graph is oriented, is actually the depth component. So that blue cloud is kind of representing, for this example, all of the area in which energy consumption is occurring. And the warmer colors are corresponding to areas where there's a lot of high energy, a lot of energy being consumed. And so we can see here from this example, there's a lot of energy being consumed in upper 20 or 30 meters of the water column. And based on what Jeremy talked about earlier, this was likely a year where they were targeting sort of those younger age classes of pollock. Next slide, please. So this sort of output is going to feed directly into question 3, which I'm going to turn it over to Yvonne to talk about.
Can everybody hear okay, I hope? And question 3 really relates to how do we relate the northern fur seals bioenergetics to pollock dynamics in a way that incorporates predation mortality on pollock and harvest rates. Next. And really shows, the models previously shown by Jeremy as part of the latest suite of ecosystem-based fisheries management tools. And we'll start with global climate models that are actually Earth-wide and are large scale, so this is like 30 to 100 kilometers. And at any given time, there are 20 to 40 models available from these ones. So we actually have to choose. We don't actually use all 20 or 40. We use them depending on their performance for sea ice. Sea ice patterns and behavior is really influential in Alaskan dynamics, and that is why it is one of our criteria to choose which models we use. And so those that perform well are the ones that we choose from. And then for the future forecasts or future scenarios, we have to make some assumptions about the greenhouse emissions. And in this case, we are choosing an ever-increasing amount of greenhouses, which is RCP 8.5, and RCP 4.5, which is a sort of median outlook that assumes that come 2100, these emissions will stabilize. These models, because they are 20 to 30, or rather 30 to 100 kilometers in scale, really need to be downscaled to a spatial resolution that is relevant both for Bering Sea dynamics and to fisheries dynamics. And so we use a downscaling regional ocean modeling system, and that is bringing everything down to 10-kilometer scale. And we can use them actually to either do long-term projections or 9-month projections, or to recreate the past. And results from these downscaled results we can actually use as input for other models. Next. So this is just a closer look at what these stacked models are. And you can see that includes at the very base the inputs for climate models, and you are modeling the ocean dynamics, the currents, the temperature, the salinity. You're including the food web base, that is the zooplankton and phytoplankton, which are really important prey for most fish. And then on top of that, we have the fish model that includes 15 species. It's a food web model driven by Bioenergetics. But importantly, this model also includes Pollock model by age and size, and it includes 16 fisheries categorized by sector, gear, and target. Next. And from each of these models that you saw, you can actually extract pertaining information to just that one model. In this case, we will be extracting information from Pollock for age 3 and older, and also for ages 0 and 1, and overlapping that with the need and availability of foraging needs and energy requirements. And the relevant portion here is that not only are we downscaling in terms of spatial resolution, 10 kilometers, but also these outputs are available daily, which means that we can follow the movements of the fish throughout the entire season, that is through June and November, how the availability changes according to the trips. And we are also able to measure the portion of the population that is actually relevant to the northern fur seals, as opposed to assuming that the entire biomass is available within their foraging range. Next. And one of the really important things is that you saw in a previous slide that pup growth really changed depending on environmental conditions interannually. So we really wanted to make sure that in addition to warm and cold years, which are relevant in Alaska because of bottom temperatures that can act as boundaries for how the fish move, we wanted to include air temperatures, storminess, winds, and other environmental and climate information that actually may be affecting either pup growth survival and or female swimming patterns. And then we can apply those variables or that information and see whether they are having an impact in the future. Next. So what does actually the future look like? Next.
Okay, so I think I'm going to jump in here first and just talk about how we're going to look at how the future looks like for northern fur seal. And to do that, we're going to be using a state-dependent behavioral model. And I'm not really going to talk about any of the specifics of the model. This is a couple of years off, and the models are quite complex. But the underlying ideas and assumptions behind these types of models are actually relatively intuitive. So the models are based on the idea that animals should act in a way to maximize their own survival and also their cumulative reproductive success. So, for example, the number of pups. A female wants to maximize the number of pups that she can have throughout her lifetime. And they're also based on this idea that animal behavior should be affected by, for example, an individual's physiological state, such as body condition, or something like their environmental state, such as where they're currently located. And so these models can actually be broken up into two primary steps. So in the first step, the inputs into this model will be all of the energetic information we're developing in earlier phases, information about how pup mass at weaning relates to the probability of pup survival, and also all of the energy landscape for Pollock. And the output of this model is, so the behavior that we're interested here is sort of the optimal location. So you can think about if the Bering Sea was gridded up into sort of 10 by 10 kilometer cells, what's the optimal cell that a female should be in given the time and a set of state variables. And so these state variables could include things like female mass, pup mass, her current location, and also the energy availability at her current location. So then what we do in step 2 is we can actually simulate a population of fur seals, and here we're just focusing on the lactating females, through time using those behavioral rules we've derived in step 1, and we're going to do that under a variety of different prey availability scenarios, which Yvonne is going to touch on in some later slides. So we've got 2 climate scenarios and 2 fishing scenarios. And the output of this step is sort of the important part. So we can really track female mass as it changes throughout the season. We can track her foraging locations, and most importantly, we can look at how those different prey availability scenarios are influencing pup mass and growth rates and the mass at weaning, so we can really understand how the population dynamics may change under the given the future availability of Pollock. Next slide.
So here, I will just reiterate that from that stacked model, from the oceanography, zooplankton, and feast, you can take information out of any individual module, and then, of course, you can run them individually up to that time. So you can run the oceanography on its own, or you can run the lower trophic levels with the oceanography, and feast, of course, runs with everything underneath it. And you can then combine the model outputs from the feast model with those of the Seattle model and from the northern fur seals to actually, build rules about how you think they're going to interact in the future. Next, please. And this is just a little bit more info on the Seattle model. Importantly, it models walleye pollock with both temperature effects on weight and how much it feeds. And from the previous slide, you can see that it actually gets zooplankton information, which is the main prey. And there are feeding interactions between walleye pollock, arrowtooth, and Pacific cod, including cannibalism. And then also this is the model that allows us to include the different fishing scenarios. And importantly, Seattle is also part of the annual stock assessment reports. So this is one way that actually information is given to the council. Next, please. And so to wrap up, what we're doing is getting new field information, both from the past, from the 1970s, all to very recent with very new recent technologies and recent field data. We are then taking spatially explicit northern fur seals bioenergetic model with separate demands of H0s and H1s, and with the older pollock, the H3, which are actually the ones that interact with the fisheries and the ones that are harvested. And then we are modeling through June to November. We are including climate, response to temperature, and prey available in all three models. And then we are synthesizing the information and putting it into a multi-species model that can include long-term fishing effects or fishing scenarios, or 9-month forecasts for seasonal or next year for scenarios, which can provide relevant information that is for ecosystem-based management and included in the SAFE reports. And with that, we'll get questions. Next, please. And we just want to thank our funding sources and contributing other projects.
Wonderful. Thank you so much for that presentation, and we will move into questions. So we do have a couple of questions so far, and folks, again, feel free to type your question into the Q&A panel if you want to ask one. The first is, what is the most recent information about the Boguslav population post-eruption?
I don't think any of us are willing to go to the island quite yet. No, but in all seriousness, for those of you that don't know, last winter, Boguslav Island began erupting, and it continued to erupt all the way through the breeding season. And several eruptions occurred during the peak of the breeding season. And the effects of this we don't know yet. There has been some flights that have gone over and taken some aerial pictures of the island in early July, which is kind of the not quite the peak of the season. Lots of sea lions and fur seals were seen on Boguslav, but we really don't have any idea how much or how those eruptions impacted the population there. We are trying to secure some funding to go next year, and hopefully Boguslav stays quiet for the next year and doesn't erupt again. So at this point, and that's a really good question, because Boguslav is an island that's been increasing quite a bit, and is kind of an example of a healthy population, and then just last year it blew up. So we hope that most of those adults either got off the island before those eruptions and have come back this year. We're not scheduled to go there this year to take a look, but possibly we'll go next year.
Thanks for that, Jeremy. Another question, and apologies if you already answered this, but what is the prey composition of animals on Boguslav compared to animals on St. Paul?
That's a really good question. Those animals on Boguslav and the animals that leave St. Paul and St. George and go into the deeper water into the basin have a completely different prey composition, and they're mostly consuming bathylagids and squids, and those have different energy content, and that in the case of bathylagids, they can be really energy-rich prey, and whereas squids are a much lower energy type prey. But yeah, the prey composition is very, very different. So on St. Paul or St. George, if they're feeding on the shelf, they're mostly consuming pollocks, capelins, herrings, various things like that.
Question 3, will you also look at models for age 0 pollock, given they are such a large part of the fur seal diet?
Yes, actually, we can look at a portion of that. FEAST actually does include age 0 pollock, and so we can look at what the overlap of the distribution is between the fur seals and the distribution of the age 0s. Importantly, you can also follow them. So that growth that Jeremy showed throughout the season of the little age pollocks growing all the way through November, that is actually included in the feast model. So that can be actually in addition to Seattle that starts at age 1.
I mean, I'll add to that. One of the things that we didn't focus in on in this talk is we will be looking at the bioenergetic, spatially explicit bioenergetics results and compare those to also the observational data that's been collected by a variety of surveys over the years. And so there's been some work that's really focused in, particularly in late August and September, the basis work looking at the distribution of age zeros. And we will be taking a look at that.
Great. So another question, how does female northern fur seal metabolic demands during lactation compare to other times of year?
Sorry, I have to unmute myself. In terms of, so I guess it's a slightly complicated, it's a good question. It's a complicated question because so the actual energy expenditure of the females themselves probably is I would imagine that it's unlikely to change that much in terms of how much energy they're spending on themselves, although I will say that we don't actually have data on metabolic rates of adult females outside of the Bering Sea. But of course, their energy expenditure or how much prey they need to take in is going to be much higher while they're in the Bering Sea because of they need to support the pup. And some preliminary output from the bioenergetic model suggests that the pup's needs can comprise almost up to about half of the female's energetic requirements, depending upon the time that you are in the season. So, for example, the pup's needs as a proportion of a female's total energy needs are going to be comparatively smaller when the pup is small early in the season, and then as the pup grows, they're going to become a larger part of the female's energy budget because her energy expenditure on her own metabolism isn't changing that much throughout the season. I hope that answered the question.
Great. So here's another one. What behaviors support the assumption females try to maximize pup production? Couldn't high fecundity and simple site fidelity be enough for annual pregnancy?
So that is those models are based on that assumption, and so yes, there could be other things that are obviously driving animal behavior, but just for this type of model that we're going to be using, that is an underlying assumption of the model. I have some experience with using these models in the past, and in my experience, they do actually do a fairly good job in reproducing the observable behavior of animals in nature, and so that's one of the reasons why sort of we chose to go down this route, is because I think, obviously we're not there yet, but they actually do a fairly good job of reproducing the patterns that we see in nature. And because there's so much information on northern fur seal behavior, we'll actually really be able to sort of ground truth the results of our model against essentially what we know that animals do in nature. So we'll be able to see how well our model compares to what we observe, which will give us an understanding of how well our model is potentially doing at predicting future female behavior.
Great, thanks for that. Here's another one. Will you be modeling the dynamics of northern smooth tongue and squid?
You know, squid is part of the food web included in feast, but we do not have reliable information so much on its movement and actual caloric content and growth. So that is very limited, and we are not including northern smooth tongue in the model. We have other small fish like herring and capelin and eulachon and myctophids, but those share a lower portion, in terms of relevance as prey for the northern fur seals. So the only one would be squid, but we really are not necessarily taking this output and putting it into the bioenergetics. The population, year-to-year variation for squids, there is actually no reliable good model to do that, and we don't have reliable biomass assessments, even from surveys or from models, to get those biomass estimates or abundance estimates, so we really have no way of anchoring that information.
Okay, so we have just 2 more questions, and then we're going to wrap it up there. So here's the last one, or the second to last one. Current Seattle model used in stock assessment process derives different ABC recommendations for pollock compared to single species models. Can you discuss the basis for this difference and effects on ABC recommendations for a fishery?
So there's different ways to set up Seattle. And Seattle model can be run in a single species mode or in multi-species mode. And relative to the current stock assessment model, when we make it as close as possible, they do perform similarly. And so we can kind of go through that model, flipping switches on. And some of those options in the Seattle model are things like accounting for changes in climate, so water temperature, changes in recruitment due to things like shifting prey for pollock. And so they're not, we include it in the stock assessment process because it provides a bit of understanding about how some of those other switches may be affecting it. But the current stock assessment model is fairly robust, and so actually they do agree more often than disagree. The value of using it in this framework is we can then look at how that multi-species-based reference point, that ABC, can also account for changes in prey needs of the northern fur seals. And so that's the idea, is to use that model to help derive those numbers that could then inform management directly.
Thank you, and this is going to be our final question. How is the project being coordinated with tribal co-managers on St. Paul or St. George?
We've been communicating with, is the mute off? Okay. We've been, Lauren Devine has been communicating with us, and our current plan is I'm going to be going to St. George here in August, and we are going to be presenting a similar presentation to the island of St. George, and we're currently trying to secure funding to go to St. Paul to discuss this project. And we are really thankful for the help that the ECO office on St. Paul has provided or offered to us, and our hope is to communicate to the individuals, or to the island communities, the project as it moves along, meaning we'll be presenting results, particularly when I'm on the islands. Right now, we've had very difficult funding resources to fly out to St. Paul and St. George, and the hope is that in conjunction with some of the research that I'll be doing, I'll be able to take some time while I'm there and communicate this project.
Thank you so much for that, Jeremy, and we are going to wrap it up now. I want to thank the research team, Jeremy, Yvonne, Kirsten, Liz. There's also Nick Bond and Karam, who are part of it too. Thank you for all of your work. Also, thank you for your willingness to work on communicating the progress of the project and share information about it. To everyone that joined, thank you. And please do reach out to us because the research team, as well as us here at LenFest, are very committed to sharing information about the project as it progresses. So thank you so much, everyone.